Welcome to the Demystifying Medicine YouTube channel. This just in. Organic chemists at UCLA have been able to synthesize the first artificial version of a promising therapeutic molecule. Found in ocean-based organisms like sea sponges, the molecule, which is known as Lissodendoric acid A, has shown potential in reducing the production of reactive oxidative species, or ROS, that are associated with brain cancers, as well as preventing the neural deterioration that causes degenerative disorders, like Parkinson's disease. ROS are a type of unstable molecule that contain oxygen and are known to react easily and cause damage to DNA, RNA, and proteins. Researchers state they accomplished this feat by using a complex 12-step reaction process, where an effective mirrored copy was produced using the often neglected and unconventional organic compound of cyclic allene. The organic chemists involved in the project anticipate that their innovative use of a temporary compound in the synthesis of a complex molecule will inspire others to use cyclic allenes to expedite the process of drug creation, as well as to challenge conventional thinking in the pharmaceutical research industry. The foundation of this research is built upon the idea that molecules can exist in two or more different forms. Called isomers, these forms tend to have the same chemical formula, but different arrangements of atoms and thus have different properties. There are different manifestations of these. The most common type of isomers are structural isomers, stereoisomers, and conformational isomers. Structural isomers have different arrangements of atoms, while stereoisomers have the same arrangement of atoms but different spatial arrangements. Conformational isomers, on the other hand, are stereoisomers that differ in the rotation around a single bond. A specific type of stereoisomers are enantiomers. To conceptualize enantiomers, just think of your hands. Enantiomers, like your hands, are mirror images of one another but are non-superimposable, meaning that they can be distinguished between. Enantiomers will look alike and have similar physical properties, but will not be the same when viewed in three-dimensional space. Because enantiomers are non-superimposable, they are considered to be chiral. This property of chirality is important, as it can affect how different enantiomers react within the body. In research settings, enantiomers should be approached with extreme caution, as they can interact with enzymes and receptors within the body in completely different ways. For example, the drug, thalidomide, which was initially used to treat morning sickness for pregnant women in the 1950s, was marketed as a mixture of enantiomers. One enantiomer provided therapeutic effects for pregnant women, while the other was found to cause birth defects. The drug was removed from the market and altered into a cancer drug in the 1960s. In a lot of cases, it may be ideal to only utilize one enantiomer. However, creating molecules in the laboratory often yields a mixture of both enantiomers. Chemically removing or reversing the unwanted ones adds difficulties, costs, and delays to the process. As such, it was a big surprise when researchers at UCLA discovered a way to harness the unique qualities of cyclic allenes to generate a very specific chiral molecule. This discovery, in turn, led to the forging of chemical reactions that produced the desired enantiomer of the Lissodendoric acid A molecule almost exclusively. This new method is both quick and efficient compared to other methods. In the past, researchers did not often use strained cyclic allenes when synthesizing new molecules due to their unstable and reactive properties, which can make them difficult to control during chemical reactions. Cyclic allenes are essentially a class of strained molecules containing ring structures, where a carbon atom shares double bonds with the neighboring carbons. In the context of chemistry, when molecules become strained, it means that they undergo bending and twisting, which increases the amount of energy between the bonds due to the repulsion of negative charges, thus altering the arrangement of atoms in a compound. So, what are the steps required to produce the desired version of Lissodendoric acid A? The researchers started off by using cyclic allene as a precursor, which is a chemical that gives rise to another chemical product. Later on, this cyclic allene precursor would lead to the production of an enantiomer or version of Lissodendoric acid A that researchers speculate could have therapeutic health effects. Then, researchers used a catalyst to reduce the cyclic allene precursor. Reduction is when electrons are gained. As a result, the precursor gives rise to a strained cyclic allene intermediate, which is a reacting species that will be consumed in later reactions. A key step in the synthesis of Lissodendoric acid A includes a cycloaddition reaction, which involves turning the double bonds into a ring structure. In the cycloaddition, 2-pyrone, a building block molecule, transforms the cyclic alene intermediate so that it forms the center of the desired molecule. A critical step in the reaction was the trapping of the cyclic alene intermediate, which helped detect the presence of an unstable intermediate that would give rise to the enantiomer that is preferred. Through the trapping step, the researchers aimed to convert the unstable intermediate to a stable form of Lissodendoric acid A. The last few steps of the synthesis included removing protecting side chain groups, which were previously used to prevent interfering side reactions and stabilize the molecule. The final step involves treating the intermediate with trifluoroacetic acid, which is a chemical reagent to remove the remaining protecting groups, thus generating Lissodendoric acid A. As of this moment, the research into the clinical applications of the molecule is still at its preliminary stage. It may take a long time until the research can be applied to human and animal models. However, the use of Lissodendoric acid A has been tested in vitro on human and mouse cell lines. In vitro testing is when tests are conducted on cells within test tubes. The test showed that Lissodendoric acid A was not toxic to humans when tested on HeLa and SK-MEL5 cell lines. Cell lines are a population of cells that can be maintained for a period of time and originate from a single common cell. The scientists also damaged mouse cancer nerve cells with a chemical known as 6-OHDA to simulate Parkinson's disease. When these cells were treated with Lissodendoric acid A, the scientists found that there was about a 50% decrease in the levels of ROS, suggesting that Lissodendoric acid A may potentially have antioxidant effects. Adding on, the scientists noticed that the acid also inhibited programmed cell death and increased the survival of cells. Lissodendoric acid A has the potential to play an important role in the body as it can provide protection against cells that can damage the fundamental building blocks in our body. These findings are important as scientists suggest that the chemical reactions to produce a version of Lissodendoric acid can be helpful in developing treatments for disorders such as Parkinson's. Parkinson's is caused by damage to cells in the part of the brain known as the substantia nigra. This brain region helps coordinate how the body moves. When these cells get damaged, this results in the quality of life issues as well as motor and non-motor issues. As the article exploring the potential of the newly synthesized Lissodendoric acid A was only published in January 2023, scientists are still working on uncovering more information about the impact of Lissodendoric acid A in animal and human models and its mechanisms on protecting cells against ROS. Overall, these studies demonstrate that cyclic allenes are powerful tools for complex molecule synthesis and should prompt the further investigation and application of these unusual compounds. If you enjoyed this video, please leave a like, comment, and subscribe. Hit the notification bell to stay updated with any future videos.